All right, welcome back. This next macromolecule is going to be proteins. So, again, have your reading guide out. We'll be going through number 9 and 10, as well as a very important one, number 11. Maybe put a star next to that one. Very, very important. And 12 and 13 as well. So, quite a bit of your reading guide is on proteins. And then you should also have this chart out. You should be on your third row. So, you should type or write protein in this last section. This next section, I should say. All right, so here we go. Proteins. So for proteins, they have a lot of different functions. A couple of key ones: structure. They definitely help with movement. They make up most of muscles. They aid in chemical reactions, such as like in the example of an enzyme. They also help in the immune system. So all kinds of different functions for proteins. A big one for this class is definitely enzymes. We actually do an entire chapter on enzymes. That'll be on your next test, not on this one. And their monomer, so that first column there, is an amino acid. And there is 20 different amino acids, and we'll get into what makes them different. For the elements for a protein, we have carbon, hydrogen, oxygen, and now we're getting into a little bit different. Remember, carbohydrates and lipids only had those three. In proteins, we also have nitrogen and sulfur. Okay, so those are also distinguishing elements for proteins. You should have that one written down. All right, now these are some functions. Now, am I going to require you to write all those in the chart? No, but I do know that it is a question on your reading guide. So you should write these down in your reading guide. I'm not going to go through each one individually, but you can always pause this. So pause this if you want to write those down now. All right, so really important. Protein is an enzyme, and like I said, we'll spend a lot of time on that in the next unit, Unit 2. But they're a type of protein that acts as a catalyst. What that means is it speeds up chemical reaction. It does that by lowering activation energy. But you can see this is kind of just aiding this process. So enzymes can be digestive, or they can break things down. They can also build things. So this example, I go back. This example here is an example of one that's breaking it down. So we have a substrate, which is sucrose, which is a disaccharide. It's going to enter the enzyme at what we would call the spot that interacts with the substrate is called the active site of the enzyme. So that, as you can see, it's in contact here. And then what are you seeing? It's breaking. Here's the bond that it's breaking. That glycosidic linkage is now gone. And now we have two monosaccharides. So in this case, this enzyme's job was to break that down. They are what we would call a polypeptide. Poly, again, meaning many. That prefix poly meaning many peptides. Many peptide bonds are what holds them together, but their polymer is basically a chain of amino acids. Again, that amino acid is the monomer. But if you ever see polypeptide, just think it's a bunch of amino acids put together. A protein might be one chain of amino acids, so it could be a polypeptide, or it could be more, and I'll explain that more in a little bit. I mentioned that the monomer is an amino acid. There is 20 different kinds of amino acid, and all these amino acids have a lot in common, but they also have some distinguishing features. So let me get into that now. All amino acids have an amino group, so that's this. And when you hear that amino, think nitrogen. Okay, when you think amine or an amino group, that's nitrogen. They also have this central carbon, and then they have what we call a carboxyl or carboxylic acid group, and that's that. So you might be, well, what makes them different? Well, what makes them different is this group down here, this R group. Okay, and that is what is different about those 20 amino acids. They have different R groups. That's also where you would get your phosphorus and your sulfur. They would be found in your R groups. Oh, sorry, not phosphorus. No phosphorus in this one. Just sulfur would be an example of that. Remember, in that elements, we said sometimes sulfur is found. Sulfur is found in that R group. And that also is known as a variable group. All right. So amino acid polymers are linked by peptide bonds. So that would be a good thing to remember. If we look here, we have 2 amino acids. Okay, they're going to be through dehydration synthesis. We're going to take water out. They're going to be linked together. Here's that bond. Okay, and then we call that, in this case, a peptide bond. Or for carbohydrates, we call that a glycosidic linkage. Now we're calling it a peptide bond for proteins. All right, conformation in proteins. If you remember back from your freshman year, we said the most important part about a protein is its shape. Well, in AP biology, yes, we still call it, sometimes we'll call it shape, but you should also be aware of this term conformation, which simply means shape. So like if you see conformation on a test, you should know that that means the shape. And that really determines its function. Shape fits function in biology. If you change the shape of a protein, it can no longer do its job. All right, so that leads us to the 4 levels of structure. This is in your reading guide, so make sure you take a look at that. You might want to write this down now. This is very, very important. These 4 levels will show up for sure, guaranteed, on your test. Primary structure is the unique sequence of amino acids. So that's all it is the sequence of amino acids, the order of the amino acids in that protein or that polypeptide. Okay? So the polypeptide's order of amino acid is its primary structure. It's all we're worried about. It's the sequence. If we move on to the secondary structure, we start to see repeated structures, repeated configurations, in other words. So if we look at these 2 that are shown in the video, we see a helix, which looks like that spiral. It's repeated spiraling. Or in the top here, we see basically it's folded, kind of like an accordion. Okay, that is called a pleated sheet. Okay, and that is a repeated conformation. So that's something unique about the secondary structure. If we look at tertiary structure, it's the overall 3-dimensional shape. Now, this would be resulting from the interactions between R groups. Okay, so we could have, that's spelled wrong, I see, but we could have like a hydrogen bond here. We could have a hydrophobic interaction. Maybe they're going to push against each other even. Here we have a disulfide bridge or an ionic bond. But all of these are interactions between R groups. Do you need to memorize the different interactions? No, but you should be aware that those different interactions are going to cause different shapes. Okay, so if we changed the R group or if we change the amino acid, you might get different interactions, and therefore that protein might be the wrong shape. So that order of amino acids is important, and it does affect this because it might change those interactions. All right, and then the last one is called quaternary structure, and that just results from more than one polypeptide units. So if we look at this diagram here and we look at hemoglobin, there's multiple polypeptides parts to that are put together, and that is what we call quaternary. So just to review, primary is simply the sequence or order of the amino acids. Secondary is those repeated configurations like pleated sheets or foldings, and those alpha helixes are basically a spiral shape. If we go back to the tertiary structure, that would be the overall 3D shape of that protein, and that's determined by those R group interactions. And then the quaternary structure then is where we have more than one polypeptide involved, put together. Okay, so those are the 4 levels of structure, and you should know those for your test. All right, just an example of how important the structure and that primary structure is. Sickle cell anemia is a disease in which one amino acid is out of order. This is thousands of polypeptides long, this protein that determines the shape of your red blood cells. And if one amino acid is ordered, we go from a red blood cell that looks completely normal, like this one, and that shape completely changed. And again, it's just because of that one amino acid change. So that primary structure does still have all kinds of impacts on the overall shape. Okay, so just be aware of that. So, protein conformation is determined by those interactions. It can also be determined by some things in the environment. Things like temperature and pH affect it immensely. So, what that can cause is if maybe an organism like a human, we are adapted to 98.6 roughly for our homeostatic mechanisms, and our proteins work best. Our proteins at 98.6, we would say, are normal. Now, maybe if we get really, really hot, this is why fever can be a problem, it can denature our proteins. Well, if our proteins don't work right, you can't live. Okay, so again, I mentioned back with, we talked about the properties of water. We mentioned how very strict our body is determining your pH in your blood. Again, pH is another thing that can cause denaturing. So that can change the shape of that protein. If you change the shape, it can no longer do its job. So those are important things to point out. All right, and that is protein. Let me just go back to that chart quickly. The monomer of a protein is an amino acid, and there's 20 of them. What makes those different is there are groups. The elements you should have are carbon, oxygen, hydrogen, nitrogen, and sulfur. Other things to keep in mind that all amino acids have an amino group, which includes that nitrogen. They also have that carboxyl or carboxylic acid group and a central carbon. For examples, you maybe have enzymes, muscles, hemoglobin, antibodies, etc. And for functions, there was 8 of them in that one slide, but maybe a couple structure, movement, chemical reactions for enzymes, things like that. Immune system would be another function. And I'm sure you have more than that as well. So that wraps up proteins.